Hi, everyone. I'm Dr. Elena Berezovska, and I'll continue to have wonderful, interesting podcasts with my colleagues, also scientists and other interesting people. And today I have a special guest, Alexander Petrenko, as my colleague, doctor, whom I met a couple of years ago in Odessa, because I know you're from Odessa. By the way, I read on your webpage, mutation.me, that you were doing a genetic test to find out your roots. And if you have result, can you share with us a little bit later this result?
Sure.
So right now, Alexander is in Austria, Vienna, because he is doing a PhD course, and he will tell about this a little bit more. And also why I was in Odessa, because I was lecturing for students of medical university, and there I met Alexander, and we had a lot of discussion on embryology, and because Alexander was like learning or studying and quite keen in embryology. So let's start a little bit more about genetic testing, but also if you can add some more words, because you have your web page with a blog and it's quite interesting. So I will provide the link to this web page under this video, and everybody can go and read in English, and also Alexander speaks in other languages as well. So I'll give you a word, just tell my our viewers a little bit more about your interest in what you're doing in Vienna, but also why we're going to talk today about very popular subject about genetic testing, especially when we're talking about IVF. So it would be like pre-implantation genetic testing. And so I'll give you a word just to tell a little bit more about yourself.
Yeah, thanks, Elena. That's my pleasure of being here today, and thanks for the inviting. I hope there in Ontario you have much better weather than we have today in Vienna, which is plus 33 and clear sky. Yeah, and thanks for these kind words. I'm a medical doctor by education, and at some point of my post-graduation career, I decided to switch to the IVF field, and not to the clinical part of that, but to the embryological part. So you know, medical laboratories, IVF, this all smart kind of microscopes, that was my passion. And I enjoyed quite a few years working there until I finally moved to Vienna and started working in scientific lab, which is focused on hepatology. So basically we're basic science or translational lab, which studies human liver disorders on the genetic level, on cellular level. And ultimately, we're trying to bring all this knowledge, all this new information we have to the clinical practice, either as providing the new tests or possibly, hopefully, new treatments. And basically, that's what we are doing. And yeah.
Now, genetics actually, it's number one, I would say, specialization in medicine that is growing in such a fast way, because we try to link genetic changes like mutation to any disease we know, and sometimes we are successful, sometimes not. But also we use genetics to find a new treatment, especially for cancer, like genetically modified treatment, and not only treatment of cancer, but many other diseases, autoimmune diseases, systemic diseases. And it's very interesting that, and I think that genetic, it's like still like in a baby condition, because it's such a huge field for investigation and learning, and no wonder that many scientists are involved in genetics and learning about genes and mutations, and also now you learning about genes that related or associated with function of liver and liver diseases and any other processes in a body that liver involves. But we will talk about liver in another podcast, and we concentrate more today on genetic testing. And I know you know a lot about IVF, especially about pre-implantation genetic testing, because we have right now some kind of discrepancy, and some doctors say like it's very useful methods that can be used like in any patient, in any women after 35 years old. Some say no, we have to do only by strong indications and not like in any kind of IVF. So what actually we know right now at the moment that is useful, what we can say to the patient, or they can hear and make some decision knowing what we know.
Oh, yeah, I mean, that's perfectly right, because genetic field overall is booming for, I don't know, for 10 years or something already.
Yeah.
And it doesn't really stop, because every year we have new and new fascinating technologies. And, you know, in the field of genetics, I feel sometimes we need this bridge of maybe scientists or genetic counselors who can actually communicate all these new findings between patients and physicians and scientists doing really smart things somewhere in the labs. And I just see how much the approach is changes itself. Because, for example, 10 years ago, when the new so-called new generation sequencing, which we maybe can cover up later, only appeared, it was quite expensive, quite not common. And nowadays it's just everywhere, from, as you mentioned, from cancer to the IVF. And that's perfectly right that especially in cancer, it's already included in the clinical guidelines. So, for example, if a patient has a biopsy for the breast cancer or for thyroid cancer, it is often that the clinical situation requires performing so-called targeted sequencing, when there is a specific panel of genes, which will then dictate whether a patient will have more susceptibility or resistance to the specific treatment. So it comes to the medicine quite in a quite fast fashion, quite fast manner, I would say. And IVF is no exclusion as well. I mean, as an optimist and enthusiast, I hope that there will be a day when the sequencing technologies will be so cheap, so available, that we'll just sequence everybody. Like neonatal screening, we will not do this by chemical tests, but if the technologies allow us, we'll just sequence every newborn baby. And that's actually a tricky question regarding this, because in the IVF, I also heard about such discussions, like whether we should do just pre-implantation genetic testing for any embryo we receive if the couple is of, let's say, advanced stage. And as far as I know, the answer from the guidelines as of now is no. And this is why we are not sure. We cannot capture all the genetic picture because our technologies are not that developed right now. So there are different types of genetic testing which we apply in the pre-implantation genetic testing. And those are, for two big topics, other big chromosomal changes, which we call aneuploidies. So for example, we suspect there's a specific embryo in a specific couple, or based on the background of this couple, might have an extra chromosome, or the missing part of some chromosome. Second big point is targeted, or let's say specific sequencing of specific genes. And I recall we had a couple from I think from Australia. Both of the partners had this variants of the genes which are associated with hemochromatosis. And that might be an issue because, for example, if one of the embryos received after IVF inherited both of the copies, that would be something we call as compound heterozygous. So basically, this disease can manifest, but we don't know how and when, and whether it will be more severe or less severe than the classical one. So in such a case, we have these embryos, we have the technology to sequence this genetic material, and we just would like to prioritize those without this both mutated copies in one. So yeah, there are many approaches, and as far as I know, the guidelines state that you should only do this in case you have specific suspicion. For example, failed pregnancies in the past, miscarriages, or maybe even failed IVF attempts. I recall there were some criteria that require you, or not require, but recommend you applying screening in case you had a few unsuccessful cycles, because even morphologically fine embryos can actually contain aneuploidies and may have an extra chromosome, for example, and that also can be linked to this issue.
Yeah, in obstetrics and gynecology guidelines, we have actually like this practical recommendation. when we use a different level of genotyping and chromosomal and karyotyping, right? And we try to explain a patient, because like it's normal when a woman or couple wants to have a healthy baby and healthy genetically and chromosomally. That's why they want to have the best embryo. And it's interesting that sometimes they require, like, they want to like know everything about embryo. And yeah, we try to explain that not always like genotyping or karyotyping will show all mutation or all kind of like problems that can affect the baby later. And also it's interesting that right now the technology is growing, like you said. Yeah, the technology now can like offer like huge varieties of genotyping, and I know it's becoming very popular, mostly on commercial level. It's like genotyping, like whole genotyping, like it's interesting that in not only us, like obstetrician gynecologists, we don't know how to interpret the data we get in, but also other doctors who dealing with these results. And when the patient brings this kind of like 3 pages description of mutation and what's the next, what we can do, because the mutation doesn't mean a disease, right? And not always like we will have a problem. But when it comes to embryo, like the couple wants to have a healthy embryo, the healthy, like, you know, and they want to be reassured that everything is fine. But again, it's a limitation. And that's why in our obstetric and gynae recommendation, actually it's a recommendation for reproductive medicine, we have kind of a very specific recommendation that we don't do like high level, like you know, like huge genotype. We don't look all chromosomes. A simple one, it's the 3 chromosomes, like looking for trisomies 13, 18, and 21, because they're most not popular, but I would say like we see them because other embryos usually with other chromosomal anomalies are dying, right? They cannot survive, but this one can survive. And this is what we do the most often. But also, like you said, we start doing more in looking for specific genes, specific mutation when it comes to diseases that are run in the families. And there are some areas in the world when actually this level of hereditary diseases are high, and the diseases could be very dangerous. But I have a question, like, you know, also there's a lot of discussion how it's safe for the fetus, embryo, because they take this test on day from 3 to 5, usually 5, right? And,
usually 5, yes.
Usually 5. And still they take in at least 2 cells, as I understand, for testing. And in a proportion to like a whole numbers of cells, it's around how many, 10, 15, 25%? Yeah.
Yeah, maybe even less, I would say, yeah.
And you know, I even know in the advance of technology and everything, I'm still thinking, okay, so my body, like, it's 100%. So if I cut my hand, it's like 10% or that. So how actually it can affect if we take the cells from embryo and what we get in the future, if it's a fact or not, and we still don't know like the true answers, right? And that's probably the biggest concern. But again, I know you were working in this area because you evaluating this, how it's safe. Actually, this testing is safe. And what can you say, like for our viewers, like should we worry or should actually we kind of like trust medicine, the science, and if it's necessary, we'll do it.
Oh, I mean, overall, that's a fantastic field, because in this field of reproductive genetics, we always have more questions than answers. And I have just this feeling that we know such a small part of the, you know, global things or mechanisms happening on this level that we cannot even prognose that. As you mentioned, right, we did in the past, so we took the biopsy of the embryo at day 3. And I know that many patients always listen to your material, so I will explain a little bit. Then the embryo is developing on the day 3, what we can do using some sophisticated microscopes, sophisticated laser, we can dissect the membrane of this embryo and take a small part of it, just containing enough genetic material to make this test. And all the rest of the embryo will be intact. And this missing part will be replaced by the dividing cells or blastomeres of other parts of the embryo. And the thing is, people don't do this that often anymore, because taking one blastomere at day 3 is still a big thing. And we definitely don't want to interfere too much with this small, almost single cell organism, exactly because, as you mentioned, we actually are not aware how it would impact. I mean, there's many babies actually born after such techniques, and we don't see any global issue with this. But nowadays it's more acceptable to make the biopsies at the later step, which is day 5 usually, or day 6 depending on the circumstances. And the technique is pretty much the same. There's sophisticated microscope, so you fix this embryo with one specific needle, and using this laser, you just dissect the membrane and take the material you need. In case of this day 5, these are not any more blastomeres, because of the different stage we take the cells, which are called trophectodermal cells. And you're absolutely right, it's actually quite a limited number of cells, because we also don't want to take too much. We don't know how much could it interfere with the development of this future fetus. And actually this might be one of the issues, because there is specific mechanism which exists in all embryos of all species, which is mosaicism. And if you're unlucky, you can just miss, for example, cells containing the mutation. But on the opposite, there are also mechanisms which are fighting with this in the natural process of development of the embryos. And sometimes we can see, I've seen, my colleagues showed me, and I've seen in some publications, how embryo extracts some parts of some cells, which then, curious people sequenced and seen that actually containing some mutations, or they are unemployed. So in some cases, embryo even itself tries to fight with this chromosomal aberrancies inside this small parts of it. Yeah, and basically what happens next, as you told, there are specific, not favorite, but most priority tests which people usually do, and which you mentioned, there are 3 chromosomal tests, 5 chromosomal tests, etc. Or even some people do so-called fish karyotyping. So basically they cover all the chromosomes to see, for example, the picture is more complicated, and they are at the stage where they are not sure what caused the issue with failed implantation or miscarriages or whatever. But yeah, these 3 chromosomal sets are quite popular, because if you miss that, if there is any chance that such baby will be developed with such chromosomal aberration, it will indeed be a burden for parents, which we might avoid if we test for these specific aberrations. Because in some cases, for example, with Down syndrome, these babies can still have a decent quality of life, they can integrate into society, but in many other cases, unfortunately, the quality of life is not that great, and this is something we as a medical community don't want to specifically cause. And also there are different techniques how to do this. I mean, what I mentioned, this test for several chromosomes, for example, there are so-called genomic hybridization technique, which uses the reference DNA, which is a DNA of a healthy person, and this test sample, which we are not aware of, and it's quite high precise. It has high sensitivity, high specificity, so people use it in different fields of medicine, including IVF. But also there is next generation sequencing, which is based not that much on the hybridization of these two reference and testing samples, but rather on the sequencing of the whole structure of the DNA of the specific. But I just have this feeling that we're not at the stage yet when we would sequence whole the genomes of these embryos. First of all, we don't have enough material often, because when we're taking these 10 cells, sometimes some cells are degrading, or sometimes it's not possible to recover all the genetic material there, or we just sample to low amount, and we will not likely rebiopsy this embryo again, because again we interfere once more, which reduces the chances of this embryo to successfully implant or even survive. But also, I mean, that's a big question of genetic medicine overall, because as I mentioned before, I'm a big fan of like, let's sequence everybody. But as you mentioned correctly, that's always a question how to interpret this data. And it's always a big issue. Then we receive this giant list of dozens or hundred potential pathogenic variants of some patient, for example. There is always a question, what was the first idea? Why we did this testing? Because if we sequence, like, for example, you or me, we will most definitely find some pathogenic variants, which might or might not be important. And that's a whole another topic. In the beginning, you asked whether I did the genetic testing already. Yes. So my wife and I received the results of this so-called ancestry testing not so far ago, a few months ago. Yeah, and it was quite funny because, you know, there are some ethnicities or some populations with known variants, so you are most likely can associate combination of some variants to some specific ethnicity, but it's not like 100%. So you cannot say that this person is only from this community, unless it's super endogenic, which is not usually the case. Yeah, and in my case, it was a lot of Finnish, Estonian, Greek, and Italian mixtures, which kind of refers to what I know about my family migration. But again, how to interpret it is always the question, because, you know, we know all this because these populations were sequenced in big studies. For example, 1,000 genomes by the UK government and the NIH. But then in countries like Ukraine, where I'm from, we don't have any large scale sequencing project, just, you know, to establish which variants we usually have in the population. So
I know this area because I even was writing articles on genetic testing for family roots, like, you know, in some scientific magazines. But anyway, I would like to mention to our viewers what is mosaic karyotyping, because some of them do not probably understand. But also when it comes to decision which embryo to transfer, then sometimes we have to transfer mosaic too. So mosaic actually means it's when we have two different karyotypes, like in cells, like one cell has this karyotype and another one has another karyotype, which is normal it's in nature. It's so we can see it quite often. But when it comes to embryo and when it comes to next step, it's like transferring the embryo. There's a question. Number one, like you said, we can miss this mosaic karyotyping because we don't check all cells. And also mosaicism could be on a different level, just like small amount of cells or quite big amount of cells. And another actually interesting question. So before, I remember like a couple of years ago, they actually didn't touch mosaic embryos because they said it's bad. Now we have a new publication that some mosaic embryos can be developed in a healthy baby. It nothing happens, like, you know, that's why you shouldn't waste mosaic embryos if you don't have any others. Of course, we prefer the perfect one, but again, the perfection of embryo, it's like very, very tricky question, because as I know, we still don't have any international classification of embryo qualification, right? So we use it visually mostly. We have some description, but, you know, and when it comes in total, we still kind of lacking of this, like I would say, universal classification of embryo gradation or embryo quality. And so a mosaic actually now we kind of use, but I have a question, because it's invasive procedure for embryo anyway, right? We cannot deny it. What is the percentage, if you have this data, if you remember, what is the percentage of when we actually lose an embryo because of the procedure? So do we have this data or not?
I mean, there are certainly some publications, but I'm not aware of any, you know, worldwide or standard percentage, because I think it's quite dependent on the clinic and the procedure. For example, there are some clinics making the mechanical biopsies, which are quite invasive, I would say. So basically, what you do using specific set of the microneedles and micromanipulators, you try to dissect like a small window in this membrane, and then to take this biopsy, which is not that much recommended nowadays. But on the opposite, not every clinic, especially in developed country, has a laser, which is quite expensive. Also, I think it depends on the embryologist performing this, because also when you operate with laser, it's a question how often do you do this, so how well trained are you? So I'm not aware about any specific percentage, but it's definitely the case. The more things you do with the embryo, the more chances to lose the embryo you have. It's just, the experienced embryologists tend to reduce this percentage to the minimum just because they do this technique so often, and it's more or less reliable inside one lab. Yeah, and also, I think you mentioned that how actually does it impact the health of the baby in the future, right? And I remember, when we don't know any specific mechanism, we always think about epigenetics. So basically, the modifications of the DNA without actually changing the structure. So, for example, switching on or off specific genes without specific mutations in these genes, so they remain intact. But for example, some methyl group, is being connected to this gene, and this is the case, for example, in cancer or in smoking-related changes in the genetic. Yeah, and I recall there were a lot of discussions regarding this, and there in my hometown there was an emergency an ICU unit, and one of the physicians told that he always knows when there's a baby after IVF, and we questioned him like, but how do you know this? Like, is there a specific phenotype? And he told no, but I think they're the most severe. But the issue is he doesn't see the healthy IVF baby or the healthy IVF babies, who underwent this genetic testing. So it's always a specific bias. And what we really need is a high scale studies. As far as I know, there were some studies, like including large cohorts of patients, like thousands of those, and there were no any major events associated with such techniques. But and again, it's a question of how deep we can go, because we cannot sequence, like, I don't know, 100,000 patients to define. Probably there are some changes, but no, according to the data we are having, it's not impacting, the quality of life, or not causing any major health complications. So, and again, it exists so many years that there are already a lot of such babies born in the world, right? And people, indeed, scientists indeed observe them, and some of them are participating in these observational studies. So far, we don't have any scary data which would say us that we should stop testing embryos for in such a manner.
When it comes to evaluation of negative effect of pre-implantation genetic testing on an embryo, it's a very tricky task, because first of all, if we transfer embryo right away, healthy embryo by karyotyping, it doesn't mean that the failure of IVF would be connected to genetic testing, or could be connected, could be by other reason. So that's why we never can predict if it's related to testing or not. Also, in those even like successful cases, when we have a healthy baby as a result of IVF, what is interesting, like, we have to follow these babies. That's why, like you mentioned, epigenetic is like, we try to figure out what actually, what kind of like, what was affecting baby when the baby was inside of the mother, and how it's actually influenced the health when the baby is adult, right? But again, it takes like 20 years at least to come to the adult age. And even if the first IVF was done in 1973, I guess, or 74. So it's still like, we don't have too much information. That's why I try to explain people and patient and everybody who's watching me or reading me, that when it comes to the success of IVF, especially negative sides of IVF, the child is protected by law, and we cannot follow this child, even if the mother, sometimes, like, some couples give a kind of like permission to follow the baby, but the majority, they don't. For many families, it's a secret. For few families, it's a struggle to conceive, and they won't forget it ever forever, because it's their child. It's like biological or non-biological, as matter, it's their child. So they don't want tell the whole world how actually was done. And it's interesting that there was a couple of years ago, I remember, during some kind of conference on reproductive medicine, some doctor, they actually wanted to create this project, project like IVF complication and and find out what's happening to the children. And they were not successful because the project cost itself a lot. You have to follow a lot of patients for many years. And why it wasn't successful, even like the majority of doctors said, oh yeah, let's do it at conference, right? But then in a couple of years, in 1 or 2 years, it's completely was forgotten. Because for many IVF clinics, for reproductive clinics, IVF, it's also a business, right? And why they have to wait for 20 years just to find out that something wrong is happening with babies, right? So it's their life at the present. And that's why in reproductive medicine, we quite often don't want to know what's happening in 20, 40 years, what happened to this baby or whatever. And it's another kind of like, I would say, dark side of reproductive medicine and medicine in general. And
that's for sure. I mean, it's not every business that even survives close to 20 years, like right in the vault.
exactly.
And if you see these pictures, 20 years ago for reproductive clinics and IVF labs, we can see totally different pictures with totally different technologies available, different microscopes, maybe less precise tools, and I can only imagine how it will look like in 20 years from now. I'm afraid even to suggest the way of our progression. But yeah, I mean, from all this story, it's a very important lesson to both patients and physicians that correlation doesn't mean causation, right? The fact that the embryo was biopsied or wasn't biopsied and then failed to implant doesn't mean that this was the cause or something. And I think this is also a fascinating field for both clinicians and researchers, this interactions between the embryos and the uterus, the endometrium, actually, right? Because there was a question I remember many years ago, like who should wait, right? Who to prioritize, the endometrium or the embryo? And I just recall, speaking of sequencing again, there was a fascinating technique which is patented, so it's not the public research or something. So some company did this many biopsies of endometrium of before and after of embryo transfer, and then from these biopsies they did something which is called RNA sequencing. So basically they took all the cells which are inside the biopsy, which are maybe 3,000 or 4,000 of cells. They extracted RNA from all these cells, and they read all this RNA molecule, because RNA ultimately is what defines which cell type are we working with or which stage of this cell we're observing. And from that they applied some machine learning tools to actually answer the question, what is the implantation window on the RNA level? And then, so they patented this technology, and then basically any lab can forward them the biopsy or the RNA, I don't remember, and they will answer like how close is to the implantation window. So you can, for example, consider that for the next transfer or for the next month, selecting the window of implantation. And there's so many things we are not aware of in this complicated process. So I'm surprised we even have such high success rates, if you're talking about even 30 or 40% of the successful cycle rates in some clinics, that's already fantastic.
The growth of reproductive medicine, especially science, it's like so huge that sometimes my brain is boiling when I'm reading what we achieved for the last 10, 15 years, especially 10 years. And what is interesting, right now we know that in 80% of cases, the quality of embryo is more important than the quality of endometrium. And that's why a lot of scientists like paying attention more on embryos. So what is the perfect embryo? Let's say the same like they're looking for perfect spermatozoa that enters the perfect egg cells.
Which is actually quite a challenging issue, because I always remember when I was working in the IVF field, I thought, what's so complex? Like we know the morphology of the spermatozoa, right? We just select the best one. But then when you sit under the microscope and you see like I don't know, hundreds or thousands of the spermatozoans, each of those are moving, and sometimes you spend a lot of time just to select this one, and to prioritize it for the intracytoplasmic injection. That's not something you can easily do.
It's miracle, or it's actually a secret of nature, because how the nature choose the perfect spermatozoid, the only one that enters the egg. And it's really interesting. But again, even like you said, we do biopsies and we know more, but in medicine, we also deny or probably do not want to deal, because we still do not understand. It's about a correspondence between uterus and actually a whole woman body and embryo. It's like two lives, right? And actually they are correspondence. They ask permission like to invade the uterus to implant. And it's interesting because we already got a lot of substances. We got like chaperones and some others, and even early pregnancy factor. So we know they correspond, they send in the signals. I'm a good embryo, please accept me. Can I just like stay here? And the same, the woman's body have to answer, okay, I'm ready to accept you or whatever. And it's interesting that we, in this area, we don't have too many kind of like answers, but it exists. And I'm hoping 10, 15 years, we will have more answers, how we actually can predict good implantation, how we can predict that this is IVF would be successful. And it will help a lot of couples, I know, because we will be more precise in getting the right answer, which embryo to transfer and when exactly to transfer, right?
Yeah,
and
that's for sure.
Yeah, and our conversation is coming to the end, and I know we can talk about this like for hours. And thank you very much for giving us such wonderful information. And I'm sure we'll continue, because, as I said, there are many other like topics even in reproductive medicine we can discuss. And thank you very much. And I hope everybody will be happy to hear our next conversation about liver. So please follow us at YouTube channel, my YouTube channel. And also don't forget to look in information about video. You will have some information about Dr. Alexander Petrenko, and you can actually follow him on Instagram, on Facebook. So thank you very much.
Thanks for having me. It's been fun.